Bismillahir Rahmanir Rahim. Today we will introduce molecular genetics. Molecular genetics deals with the study of the molecules and processes whereby genetic information is stored as a code, genetic code, which is a sequence of deoxyribonucleotides in DNA. This sequence of ribonucleotides is transcribed or copied into a sequence of ribonucleotides in messenger RNA. This code is then decoded or translated from a sequence of ribonucleotides into a sequence of amino acids in a protein. This protein, either directly or by acting as enzymes forming other molecules, lead to the expression of a trait such as the skin color, in which melanin is synthesized in the skin to give its color due to the presence of specific enzymes that synthesize this melanin. These processes are described by what is called the central dogma. The central dogma described how a gene is ultimately expressed. A genetic information is stored in DNA genes in genome and chromosome as a sequence of deoxyribonucleotides ATC, GG, and so on, and the DNA is formed of two complementary strands, the anti-sense strand and the sense strand, and they are complementary so that A is complementary with T and T with A, C with G, and so on. Now, the anti-sense strand is used for the transcription into messenger RNA, which is complementary to the anti-sense strand by formation of sequence of ribonucleotides. The sequence of ribonucleotides in messenger RNA is similar to that of the sense strand, except that U replaces T. And this sequence, which represents the genetic code in the gene, is transported into the cytoplasm of the cell, where each three nucleotides act as a codon, which is translated into an amino acid. So the sequence of nucleotides in messenger RNA is translated into a sequence of amino acids in a polypeptide or protein. This polypeptide or protein is folded in order to form the structural and functional protein, such as the cystic fibrosis transmembrane conductance regulator protein, CFTR, which is synthesized in the lining epithelium of pulmonary airway airways. Pulmonary airways, the epithelial lining of these airways, such as the bronchi and bronchioles, is coated with a mucus coating which traps the pathogens and foreign substances and has to get rid of it to the outside. The epithelial cells form or synthesize CFTR proteins, which are embedded in the membrane of these cells, and they help in transport of chloride ions to the surface mucus of these cells, and it traps the water molecules, forming a hydrated mucus, which is easy to move over the surface of the cells by the mucociliary clearance, which carries the pathogens and foreign substances out of the airways, leading to a healthy life. Now, in the case that CFTR genes are mutated, the transcribed and translated proteins result in the formation of deformed CFTR proteins, which cannot pass or cannot transport chloride ions, and so the mucus will not be able to be hydrated, and so it will plug the airways and containing the pathogens and bacteria, and resulting in the signs of cystic fibrosis.